Listener needs to hold on to today. It is this: structure predicts behavior.
Structure predicts behavior.
Exactly. We aren't just memorizing parts of a cell for a test. We are looking at how the physical layout of a bacterium, its walls, its motors, its sensors, determines exactly how it attacks us. And, conversely, how that same structure dictates whether our medicines will destroy it or just bounce right off.
It's the ultimate form versus function discussion. And to set the stage, the source material opens with this really compelling comparison that I think perfectly frames the stakes. It contrasts the minimalist system with the maximalist system.
This is such a crucial concept for understanding biological fragility. On one side, you have the maximalist system, that is us. Specifically, think about a human neuron.
The brain cell.
Right. A neuron is, it's arguably the most complex high-performance machine in the known universe. It facilitates consciousness, memory, rapid signaling. But being a maximalist comes with a heavy price tag. It is incredibly expensive to maintain, and it is incredibly fragile.
High maintenance, high risk.
Precisely. A neuron needs a perfect environment. It needs exact temperature control, specific ion balances, constant oxygen, and protection from any kind of physical shock. If you look at a neuron the wrong way, it dies.
And on the other end of the spectrum, you have the bacterium, the minimalist.
The bacterium is the ultimate survivalist. It is stripped down to the absolute bare essentials required for life. No frills, no luxury features, just a rugged chassis, an engine, and a replication code.
It's the difference between a Formula One car and a tank.
You can do
Or
it.
maybe, I don't know,
Let's
a
work
cockroach.
That
today
tank is
to
a good
make
analogy.
tomorrow a better
And this
day.
comparison explains a phenomenon called collateral
You can
damage
do
and
it.
infections.
Let's work
The source
today to
material
make
points out that
tomorrow a
often
better
the things our body
day.
uses to fight bacterial-like inflammation,
You can do
fever,
it.
oxidative
Let's-
stress, they actually hurt our own maximalist cells more than they hurt the minimalist bacteria.
Because the bacteria are built to weather the storm, but our delicate neurons just aren't.
Exactly. So often, the damage from a disease isn't the bacteria eating you. It's your body burning down the house to kill a guest that is, for all intents and purposes, fireproof.
That is a terrifying image, but it really highlights why we need to understand the enemy's architecture. We need to know what makes them so fireproof. So let's unroll the blueprints. If we were to build a generic bacterium from scratch, what's in the starter kit?
The sources refer to this as the shared core. These are the universal features you find in essentially all bacteria, whether it's the E. coli in your gut or the staph on your skin.
Okay, walk us through the chassis.
It starts with the plasma membrane. This is the barrier that defines inside versus outside. It holds the cytoplasm, which is sort of the factory floor, a gel-like fluid where all the metabolic work happens. But the real magic is what's floating in that fluid, specifically the DNA and the ribosomes.
Let's talk about the DNA first. In human cells, we treat our DNA like the crown jewels. We lock it inside a heavy vault called the nucleus.
Right. We protect the code at all costs. But bacteria, they don't have a nucleus. They are prokaryotes, which literally means before the nut, or, before the nucleus. Their DNA is just floating right there in the open cytoplasm in a tangled clump called the nucleoid.
That seems risky. Why leave your instruction manual out in the open like that?
Speed. It's all about speed. Because the DNA is accessible, the machinery that reads the code and builds proteins can jump onto it immediately. They don't have to wait for the code to be copied and shipped out of a vault like we do. They can replicate and react to their environment almost instantly.
So while our cells are still filing the paperwork to build a protein, the bacteria has already built 1,000 of them.
Exactly. It's a trade-off. Less protection, but maximum velocity.
Now, the other critical piece of machinery you mentioned is the ribosome, the protein factory. The notes make a very big deal about the specific specs of these factories.
This is one of the most important numbers in medicine, 70S.
70S.
Okay.
That's the classification for bacterial ribosomes.
Yes. The S stands for Svedberg units. It's a measure of how fast a particle settles in a centrifuge, which is basically a proxy for size and density. Bacteria have 70S ribosomes. Humans, on the other hand, have 80S ribosomes.
It sounds like a small difference, 70 versus 80.
Numerically, sure, but architecturally, they are very different shapes. Imagine a lock. The bacterial lock, the 70S one, has a different keyhole than the human lock, the 80S.
And this is the foundation of antibiotic therapy.
It is the antibiotic logic. We can ingest drugs like tetracycline or erythromycin because they are designed as keys that only fit the 70S lock. They gum up the works of the bacterial factory, stopping protein production, but they bounce harmlessly off our own 80S ribosomes.
Selective toxicity. It's a sniper shot, not a carpet bomb.
Ideally, yes. Though, as we will see later, there are some evolutionary ghosts that complicate that picture a bit, but
Okay.
fundamentally, the 70s ribosome is a prime target because it is distinct from us.
So we have the core, membrane, cytoplasm, open source DNA, and 70S ribosomes. But the source material lists a whole catalog of optional extras.
And this is where the story gets darker. The core makes the bacteria alive. The extras make them dangerous. These are the components that vary between species and determine if a bacteria is a harmless passenger or a lethal pathogen.
And the notes point out that bacteria are masters of redox. What does that mean in this context?
It refers to their metabolic flexibility. It's another optional extra in terms of capability. If you cut off oxygen to a human brain, the neurons die in minutes. We are obligate aerobes. But bacteria, many of them can switch gears instantly.
No oxygen, no problem.
Exactly. They switch to anaerobic respiration or fermentation. They can use sulfur, nitrogen, or even iron to breathe. This flexibility allows them to survive in deep wounds in the gut or in abscesses where oxygen can't reach. It's part of that rugged, minimalist design.
Let's move to the most prominent extra on the list. The source material puts the spotlight squarely on the cell wall.
The cell wall is the star of the show. If you take nothing else away from bacterial anatomy, you have to understand the wall.
Why is it so critical? I mean, our cells have membranes. Why do bacteria need this heavy-duty wall on top of that?
It comes down to physics, specifically pressure. Inside a bacterium, the concentration of salts, sugars, and nutrients is incredibly high because nature abhors a vacuum, or in this case, a gradient water, wants to rush inside the cell to balance it out.
Osmosis.
Right. And this creates immense outward pressure called turgor pressure. We are talking about 2 atmospheres of pressure inside that tiny cell.
That's the same pressure as a car tire.
Exactly. So a bacterium is basically an overinflated tire. If they only had that flimsy plasma membrane, they would pop instantly. They would explode. We call that osmotic lysis.
So the cell wall is the tire tread. It's the corset that holds everything together.
Correct. It provides rigidity and structural integrity. And the material it is made of is unique to bacteria. It's called peptidoglycan.
Peptidoglycan. Okay, let's unpack that word because it sounds like a mouthful, but it describes the structure perfectly.
It does. Peptido refers to peptides, which are short chains of amino acids protein fragments. Glycan refers to sugars. So visualize a lattice. You have long chains of sugars running parallel to each other, and they are stitched together crosswise by these protein staples.
It's chain link fence.
A 3D, rigid chain link fence wrapping the entire cell. And here's the medical jackpot. Humans do not make peptidoglycan. We don't have it. We don't need it because our cells don't live under that kind of pressure.
So if we can find a chemical that breaks that fence,
we can kill the bacteria without touching a single human cell. This is how penicillin works. It doesn't dissolve the bacteria. It breaks the tools the bacteria uses to build the crosslinks in the fence. The bacteria keeps growing, the wall gets weaker, the internal pressure takes over, and
it blows itself up. That is elegant.
It is. And in microbiology, there is always a but. Not all cell walls are built the same way. This brings us to the great divide.
Gram-positive versus gram-negative. I remember this from high school biology, the purple stain versus the pink stain. But I never really understood the architectural difference.
It is the most fundamental split in the bacterial world. Imagine you are an architect building a fortress. You have 2 design philosophies. Design A is the gram-positive approach.
Okay, what does the gram-positive fortress look like?
It relies on sheer thickness. It builds a massive monolithic wall of peptidoglycan. We are talking 40, 50, maybe 80 layers thick.
So it's a castle keep with 10-foot stone walls.
Exactly. And embedded in that wall are these structural rebar beams called teichoic acids. They give it extra stability. Because that wall is so thick and sponge-like, when we use the crystal violet dye in the lab, the wall soaks it up and holds it tight. That's why it stains purple.
Okay, thick wall, purple stain. That's gram-positive. What is the alternative? Gram-negative.
Gram-negative is a completely different strategy. Instead of a thick wall, they have a very thin layer of peptidoglycan, maybe only 1 or 2 sheets.
That sounds weaker.
You would think so. But they compensate by adding a feature the gram-positives don't have. They have a second membrane, an outer membrane.
So it's a sandwich. Inner membrane, thin wall, outer membrane.
Exactly. They are wearing an armored hazmat suit over their thin wall. And this outer membrane is a game changer for medicine.
The source calls this intrinsic resistance.
Yes. Think about it. If you are a drug molecule and your job is to attack the peptidoglycan wall in a gram-positive bacteria, the wall is right there. It's exposed. You can walk right up to it. But in a gram-negative bacteria, The wall is hidden behind that outer membrane.
You have to get past the security guard first.
And the security guards are strict. The outer membrane has channels called porins.
Porins, these are like gates.
They are gatekeepers. They are water-filled channels that allow nutrients in, but they are very specific about size and charge. If an antibiotic molecule is too big, like vancomycin, which is a huge molecule, the porin just says, nope, you don't fit.
The vancomycin, which crushes gram-positive infections, is useless against gram-negatives simply because it can't fit through the door.
Precisely. It bounces off the armor. This is why gram-negative infections like E. coli, Pseudomonas, Salmonella are generally much, much harder to treat. They have better perimeter security.
But the outer membrane isn't just a defensive shield, is it?
Right.
The source material takes a pretty dark turn here. It introduces a component called LPS.
LPS, lipopolysaccharide. If the cell wall is the shield, LPS is the booby trap.
The outline links LPS directly to immune catastrophe and septic shock. Walk us through this. What is LPS physically?
Physically, it is the material that makes up the outer face of that outer membrane. It's a hairy-looking molecule sticking out into the world. But the dangerous part is the root, buried deep in the membrane. It's called lipid A.
Lipid A. And this is a toxin.
It is an endotoxin. That distinction matters. Some bacteria actively shoot poisons at you. Those are exotoxins. But LPS is structural. It is part of the bacterial body. The bacteria isn't using it as a weapon. It is the bacteria.
So why is it so deadly?
The trouble starts when the bacteria dies or divides. Imagine you have a patient with a gram-negative infection in their blood. You give them a strong antibiotic. It works. It kills the bacteria. The cells burst open.
And all that lipid A is released into the bloodstream.
Exactly. Now, your immune system, specifically your macrophages, patrols the blood. When they detect lipid A, they don't just see a foreign object, they recognize it as a maximal existential threat. They panic.
It's a 5-alarm fire.
They release massive amounts of signaling chemicals called cytokines. This is the cytokine storm. These signals tell your blood vessels to dilate, to widen, to let more immune cells rush to the scene.
But if that happens everywhere in your body at once,
your blood volume effectively disappears because the vessels get too big, your blood pressure crashes, your organs, kidneys, liver, brain stop getting oxygen. That is septic shock.
so the antibiotic killed the bacteria, but the structural debris from the bacteria triggered the immune system to crash the entire system.
That is the tragic irony. Septic shock isn't the bacteria destroying your organs directly. It is an immune catastrophe. It is friendly fire on a massive scale.
There was a specific neuroscience hook in the notes about LPS that I found absolutely fascinating. It connects this gut or blood infection to the brain.
This is really cutting-edge understanding of sickness behavior. We used to think that for a bacteria to affect the brain, it had to physically cross the blood-brain barrier and infect the brain tissue.
But LPS breaks that rule.
It bypasses it. Let's say you have a urinary tract infection. The bacteria are in your bladder, nowhere near your brain. But the LPS is triggering those cytokines in your blood. Those cytokines travel to the brain. They can't cross the barrier easily, but they can knock on the door.
They signal across the barrier.
Right. They activate the brain's own immune cells, the microglia. The microglia go on alert and start changing the neurochemistry of the brain. They induce lethargy, fever, loss of appetite.
That feeling of, I just want to curl up in a ball and not move.
That is a biological program. That is your brain forcing you to rest so you can heal. But in elderly patients, or if the signal is too strong, this inflammation causes delirium and acute confusion.
So when an elderly relative suddenly gets confused or starts hallucinating, it might not be a stroke or dementia, it could be a UTI.
It is extremely common. The confusion is the architectural echo of the LPS molecule reverberating all the way to the neural network. Structure predicts behavior.
That is incredible. Okay, we've covered the walls and the toxins. Now let's talk about stealth. The notes describe an invisibility cloak.
The capsule.
This sounds like sci-fi, but it's just biology. What is a capsule?
It's exactly what it sounds like. Some bacteria, like Streptococcus pneumonia, secrete a thick, slimy, sugary goo that coats their entire outer surface. It covers the cell wall, the membrane, everything.
And what is the strategic advantage of being covered in slime?
It prevents phagocytosis.
Phagocytosis, that's the Pac-Man act, where our immune cells eat the bacteria.
Right. Neutrophils and macrophages need to physically grab a bacterium to eat it. They need surface friction. They need a grip. But the capsule is slippery, it is hydrated and slimy.
So the immune cell tries to grab it, and it just squirts away.
The classic analogy is trying to catch a greased pig, or a wet bar of soap in the shower. The immune cells just can't get a handle on it.
The source notes mention the spleen specifically here. Why is the spleen important for these greased pigs?
The spleen is the body's primary blood filter. It's a meshwork that traps bacteria physically, holding them in place so the immune cells can finally grab them. It's our best defense against encapsulated bacteria.
So if you don't have a spleen,
If you've had a splenectomy, maybe from a car accident or disease, you are in really trouble. You've lost the physical trap. Encapsulated bacteria that a normal person would fight off easily can kill a person without a spleen in hours.
because they have the invisibility cloak and no net to catch them.
Exactly. It changes the medical management completely. You have to vaccinate these patients aggressively to teach their immune system to recognize the slime coat.
Let's look at the other appendages on the list. We've got the stealth cloak, but we also have engines and grappling hooks, flagella and pili.
These are the tools of interaction. Flagella are for movement, and I don't want you to picture a tail wagging back and forth. This is a rotary motor.
A motor, like a machine.
Literally. It has a rotor, a stator, and a drive shaft embedded in the cell wall. It spins at thousands of RPM. It acts like a boat propeller.
And they use this to hunt. The notes mention chemotaxis.
Bacteria aren't just drifting aimlessly. They can sense chemical gradients, sugar, amino acids. If they sense food, the motor spins to push them toward it. If they sense a toxin, they tumble and swim away. It is purposeful, directed movement.
And the pili, or fimbriae.
These are different. They aren't for swimming, they're for sticking. Think of them as Velcro hairs covering the surface.
If a bacterium can't attach, it can't infect.
That is the golden rule. Think about the physical forces in your body. Your bladder flushes urine. Your gut pushes food through with peristalsis.
You can do it.
Your lungs
Let's
cough up
work
mucus.
today to make
Everything is trying to
tomorrow a
expel
better day.
intruders.
So the
Okay.
bacteria need to hold on for
You
dear
can
life.
do it.
Pili are the
Let's
grappling
work
hooks.
today to
E.
make
coli
tomorrow a
uses
better
them to
day.
lock onto the cells of
Okay.
your bladder so they don't get
You
flushed
can
out.
do it.
Neisseria
Let-
gonorrhea uses them to hold onto the reproductive tract. If you knock out the pili, the bacteria becomes harmless because it just gets washed away.
The notes also mention a specific type of pili involved in conjugation, the sex pilis.
Yes, this is how bacteria share secrets. A bacterium can extend a special pilis, latch onto another bacterium, and reel it in. Once they are touching, they open a channel and copy a piece of DNA to send across to the neighbor.
And what kind of information are they swapping?
Usually it's the good stuff, survival manuals. Often they are transferring plasmids, small loops of DNA that contain antibiotic resistance genes.
So one bacteria figures out how to beat penicillin, and it texts the instructions to everyone nearby.
It's peer-to-peer file sharing for superbugs, and it can happen between different species. It's a major driver of the resistance crisis we are facing.
So far, we have talked about the bacterium as a lone wolf. It has its armor, its motor, its grappling hooks. But the source material makes a pivotal point in Section 6. Bacteria rarely work alone. They build cities.
This is the concept of the biofilm, and honestly, this changes everything about how we treat infection.
The outline defines it as a structured community with an extracellular matrix. Break that down for us.
In the lab, we study bacteria floating in liquid. We call that planktonic growth. But in the real world, in the body, bacteria prefer to find a surface, attach, and then secrete a matrix.
A slime city.
Essentially, they build a dome over the colony made of sugars, DNA, and proteins. This is a biofilm. Plaque on your teeth is a biofilm. The gunk that clogs a catheter is a biofilm. The infection in the lungs of a cystic fibrosis patient is a biofilm.
Why is this structure so much harder to kill than individual bacteria? The source uses a distributed network analogy.
It's brilliant. A biofilm isn't just a pile of cells, it's organized. There are water channels to bring in nutrients. But the key is that the bacteria behave differently depending on where they are in the city. The ones on the outside are active, but the ones deep inside,
What happens to them?
they are cut off from oxygen and food, so they go into a state of dormancy. They slow their metabolism down to almost zero. We call them persister cells.
And why is a sleeping bacteria a problem?
Because antibiotics target activity. Think about it. Penicillin attacks the building of the cell wall. Tetracycline attacks the active production of proteins. If the bacteria isn't building or producing...
The drug has nothing to attack. It's invisible.
Exactly. The drug kills the active guys on the surface. You feel better. You stop the drug. But the sleeper cells deep in the bunker are untouched. Once the antibiotic washes out, they wake up, repopulate the city, and the infection comes roaring back.
This explains chronic infections. Why some people just can't shake a UTI or a sinus infection.
Biofilm leads to chronic infection, which leads to drug failure. That is the logic chain. It's not genetic resistance. It's tolerance based on lifestyle. It's a siege. And right now, our best weapon against biofilms on medical devices is often just to remove the device. You have to physically tear down the city.
We've covered a massive amount of architecture, but I want to circle back to one technical detail we touched on at the very beginning, the ribosome. We said the 70S versus 80S difference allows us to take drugs that kill bacteria but not us. But the notes have a safety warning here.
Yes, this is a plot twist billions of years in the making.
It involves mitochondria.
It does. We know that mitochondria, the power plants inside human cells, have a strange origin story. The endosymbiotic theory suggests that way back in evolutionary history, a large primitive cell swallowed a smaller bacterium.
And instead of digesting it, they became roommates.
They became partners. The small bacteria provided energy, the big cell provided protection. That small bacteria evolved into our mitochondria.
So deep down, our mitochondria are basically domesticated bacteria.
Correct. And because of that heritage, mitochondria still have some of their own DNA and, crucially, their own ribosomes.
And let me guess, those ribosomes look familiar.
They look remarkably like the bacterial 70S ribosomes.
I see the problem. We take a drug to attack the 70S bacterial ribosomes.
And if the dose is too high or the patient is susceptible, the drug can get confused. It starts inhibiting the ribosomes inside your own mitochondria.
Friendly fire again.
It shuts down your own power plants. This is why certain antibiotics, like aminoglycosides gentamicin, can cause specific side effects like deafness or kidney damage. Those tissues are energy hungry. They rely heavily on mitochondria. When the drug knocks out the mitochondrial ribosomes, the cells fail.
It's a reminder that we aren't as distinct from bacteria as we'd like to think. We are maximalist systems built on a minimalist foundation.
We have a little bit of the enemy inside us.
Okay, we're coming into the home stretch. We've covered the blueprint, the armor, the weapons, and the cities. I want to do a rapid-fire dispelling myths segment based on the source material. I'll throw out a common misconception. You give me the architectural reality.
Well, let's do it.
Myth number 1, all gram-negative bacteria are deadly killers.
False. Reality. Structure predicts potential risk, not destiny. You have gram-negatives like E. coli in your gut right now helping you digest food. They have LPS, but it remains contained. The danger is about context. E. coli in the gut is a friend. E. coli in the blood is a crisis.
Myth number 2, antibiotics work instantly to end the threat.
False. Reality, the death rattle can be dangerous. As we discussed with LPS, killing a bacteria effectively blows them up, sepeliosis releasing toxins. A patient can actually get physiologically worse, lower blood pressure, higher fever in the first few hours of treatment as those structures break apart.
Myth number 3, capsules are just optional decorations.
False. Reality. Capsules are stealth technology. A bacterium with a capsule is exponentially more virulent than the same bacterium without one. It completely changes the rules of engagement with the immune system.
And finally, let's recap the logic chains. If the listener is going to lock 3 concepts into their memory today, what are the causal links?
Chain 1. Gram-negative structure, that's the thin wall plus outer membrane, leads to LPS. LPS leads to cytokine storm, which leads to septic shock. Chain 2, capsule leads to decreased phagocytosis because it's slippery, which leads to increased virulence or survival. And chain 3, biofilm leads to metabolic dormancy, which leads to antibiotic tolerance and chronic infection.
It really does all come down to the architecture.
It's geometry. Medicine is essentially applied geometry. Does the drug fit the lock? Does the immune cell grip the surface? It's a game of shapes.
The source ends with a quote that I think sums this up perfectly. Bacterial disease is not about size or strength. It's about architecture meeting immunity.
I love that quote. It frames the struggle correctly. It's not a boxing match where the biggest guy wins. It's a lock-picking contest. It's about who has the right key for the right door.
And that leads us to our final thought for the day. We started by comparing the maximalist neuron, the fragile masterpiece of human evolution, to the minimalist bacteria, the rugged survivor.
Right.
We rely on our immune system and our technology to keep us safe. But bacteria are evolutionary speedrunners. They can change their architecture. They can thicken their walls, alter their porins, hide their antigens.
They can iterate a new design in 20 minutes. It takes us 20 years to reach adulthood. They adapt at a pace we can barely comprehend.
So the question to leave you with is this. In an arms race between a complex static fortress, which is us, and a rapidly shifting minimalist shapeshifter, which is them, how long can our static drugs keep the advantage? If structure predicts behavior and they can change their structure, can we keep up?
That is the billion-dollar question keeping microbiologists awake at night.
Something to chew on next time you reach for the hand sanitizer. Thanks for diving deep with us today.
Always a pleasure.
We'll catch you on the next deep dive. Stay curious.